Hello all. Today I, Sumant Jangam, and my teammates Clarice Romi and Aishwarya EK are here to present the research paper titled Harpin Hpa1 Promotes Flower Development in Impatiens and Parochetus Plants. It was being published in the Botanical Studies Journal by Liang Dong et al. Let's dive into the introduction. Harpin Hpa1, a protein produced by Xanthomonas oryzae, has been known for its beneficial effects on plant growth, development, and defense responses. The 10-42 residue fragment of Hpa1, known as Hpa1 10-42, was found to be more effective. Its potency ranges from 1.3 to 7.5 times greater than Hpa1 across different plant species like Arabidopsis, rice, tea, and wheat. But it shows variation in effectiveness across different plant species. For instance, in rice, Hpa1 10-42 residue is notably more effective, being 2.7 and 7.5 times stronger than Hpa1 in eliciting resistance to blast and bacterial blight, respectively. But in tobacco, it is less potent than Hpa1 in eliciting the hypersensitive response. This indicates that the bioactivity will differs in different plants. The aim of this experiment was to explore the impact of Hpa1 and Hpa1 10-42 residue on flower development in impatiens and parochetus plants, with a focus on enhancing ornamental value through improved flower growth, delayed senescence, and increased anthocyanin concentration.
And now we can discuss about the materials and methods. In their studies, they used E. coli cells as the PET30A positive vector for synthesizing the protein. And to ensure the purity, the proteins were fused with a his tag, facilitating purification through the nickel chromatography with the elution using aqueous imidazole solutions. The highly purified HPA1 his protein was obtained from the eluent containing 200 millimolar imidazole, followed by the removal of his tag using the Norwegian endokinase cleavage capture kit. And the protein concentration were determined with appropriate dilutions made for HPA1 and HPA142 preparations and the supplementation with bovine serum albumin for EVP preparations. And the final concentration of all protein preparations was standardized at 10 microgram per milliliter and supplemented with a surfactant Silvet 77 for uniform distribution on the plant surface. And the bioactivity of these proteins was then confirmed through the induction of HR in leaves of tobacco. And in the study, we used empty vector plasmid sample as a negative control. The plant treatment and flower observation. They studied the growth of 4 types of plants: Parochetus communis, Impatiens balsamina, Impatiens platypetala, and Impatiens valeriana. They put them in a growth chambers with specific conditions for temperature, humidity, and light. After 10 days, they sprayed them with protein solutions. And over the next month, they watched how their flowers developed and when they started to fade. They looked at 300 flowers from 30 plants across 3 experiments to see how each plant and treatment combinations affected the flower growth. And they measured the flower diameter before and after the treatment to see how they changed over time. The anthocyanin measurement. The extraction was performed by homogenizing the fresh flowers in liquid nitrogen and lyophilizing the powders. The anthocyanin was extracted by incubation in methanol solution containing 1% hydrochloric acid. And the concentration is quantified by spectrophotometry. The data analysis. The experiments were conducted using a completely randomized design and repeated at least 3 times. And the quantitative data was analyzed using IBM SPSS 19.0 software and the homogeneity of variance determined by Levin test. The data distribution pattern confirmed by Kolmogorov-Smirnov test and PP plots. And the analysis of variance performed along with Fisher's test, significant differences in test and Turkey's Kamas test.
Now let's discuss the results of the experiment. First, we can discuss about the bioactivity assessment result. When the HPA and HPA1 10-42 residue histidine fusion protein as well as the EVP sample was loaded onto the gel and was subjected to TSDS page, it was observed that the EVP sample did not form any kind of band on the gel, whereas the HPA1 and its short length fragment they formed a band on the gel. This means that the EVP sample contain inactive protein while the HPA1 and its short length fragment they contain the active form of the protein. Later on, the hypersensitivity response induction assay was done. This revealed that the HPA1 and HPA1 10 to 42 residue, they were active once the histidine tag was removed from the protein. When the tobacco leaf was treated with the HPA1 as well as its short length fragment, it was observed that both this protein formed some kind of necrotic patches on the leaf, whereas the EVP sample does not cause any kind of necrotic patches on the leaf. Now let's see the effect of the harpin protein on the flower growth and flower maintenance period. The developmental stages of the flowers, that is the impatient species as well as the Parochetus communis, can be categorized into 6 stages, starting from the S1 stage to the S6 stage. The S5 stage is actually the fully bloomed flower stage, and S6 stage is the stage when the flower starts to deteriorate. And the transition stage from S5 to S6 is called florescent period or the maintenance period. This is the time when the flower remains fully bloomed and remains in a fresh state. So the plant was treated with the EVP sample and HPA1 and its short-length fragment protein. When the plants were treated with these samples, it was observed that in those plants, the treatment was given using the harpin proteins. There was a shortening of the transition time from S1 to S5 stage, which means the flower bud takes less time to fully develop into a bloomed flower. But the transition time from S5 to S6 got extended, which means the fully bloomed flower remains in a fresh state for more days when compared to those plants which were treated with EVP sample. This graph explains the same. If you look into this, Impatiens balsamina, we can see that it takes about only 7 days. It takes only 7 days for the fully bloomed flower to get deteriorated. That is, the transition takes place from S5 to S6 stage within 7 days. But when you compare it with the plants which were treated with the HPA1 and HPA1 10 to 42 residue fragment, they have a more life than those plants with the which were treated with EVP sample. And again, if you look the transition stage from S1 to S2 stage, those plants which were treated with EVP sample had a more time to change from one phase to another phase. But in those plants which were given treatment using the harpin protein, they take much lesser time to change from one stage to another stage. Now, the effect on anthocyanin content. It was observed that the harpin can increase the anthocyanin content of the flowers also. This is clearly depicted in the graph which is given here. In all the 4 species of the plants which were given treatment using the EVP sample and HPA1 sample as well as the HPA1 10 to 42 residue fragment, it was observed that the EVP sample had a lesser anthocyanin content when compared to the plants which were given treatment using the harpin proteins. And when you check the graph, you will also understand that in those plants which were given treatment using the HPA1 10 to 42 residue fragment, they showed the more increase in anthocyanin content when compared to the other 2. So this means that the short fragment length was found to be more efficient than the long length fragment. These are the images of the impatiens plants and the Parochetus plants which the researchers have used for their work. It can be clearly understood from the picture that in the untreated plant as well as the plant treated with EVP sample, the anthocyanin pigment concentration was very less. So the flower has a light color when compared to the plants which were treated with HPA1 and HPA1 10 to 42 residue. So these flowers are more brighter in color and they have a higher concentration of anthocyanin. And it's also being said that the anthocyanin content was found to be very high during the S5 stage, that is the fully bloomed stage. Here there is also the images of the Parochetus plant which they have used for their study. And here you can see that in the plant which was given treatment using HPA1 10 to 42 residue, the flower had a more brighter flowers when compared to the other 3. In HPA1 also the flowers appear to be more brighter because of the increased anthocyanin concentration when compared to the untreated and EVP sample. But HPA1 10 to 42 residue was found to be more efficient in increasing the anthocyanin concentration than the HPA1. Now, this is a graph which explains how the treatment has affected the flower diameter or the flower opening in the plants which were given treatment, as well as the plants which were not treated with harpin protein. From the graph, it's quite clear that in the plants which were given treatment using the harpin, there was an increase in the flower diameter when compared to the plants which were given treatment using the EVP sample, especially in those plants which were given treatment using the shorter length fragment protein. The flower diameter was found to be very high when compared to the plants which were treated with EVP sample.
Now we can move to the discussion part. The aim of the study was to assess the effects of HPA1 and HPA1 10 to 42 on flowering properties of Paracetus communis, Impatiens balsamina, and Impatiens platypetala. The hypothesis was that the proteins would enhance the flower growth and prolong maintenance period by improving the ornamental qualities. And the results also confirms the hypothesis, showing both proteins promoted the flower growth, delayed senescence, and increased anthocyanin levels by enhancing the ornamental appearance.
Impatiens and Parochetus plants chosen for their suitability as models for studying flowering properties. Effects of HPA1 and HPA1 10 to 42 residue significantly improved flower qualities, suggesting increased ornamental value. HPA1 10 to 42 residue appeared more effective than HPA1 in promoting flower growth and extending maintenance period. Molecular mechanisms underlying effects were not investigated, warranting future research. Understanding physiological and pathological responses is crucial for uncovering roles of harpins in plant development and immunity. Further studies needed to elucidate genetic and molecular mechanisms of HPA1 and HPA1 10 to 42 residue in ornamental plants like Paracetus communis, Impatiens balsamina, Impatiens platypetala, and Impatiens valeriana. Thank you.